Hi, good afternoon, and welcome to the 5th webinar organized by GFB Onlus. So first of all, I want to thank all of you to attend this webinar series where we try to bring this the knowledge of the current clinical programs to our community. And I want to thank also the people that is attending in this webinar, our international patients, but also today we have some representation of the university community from Rome and from elsewhere. So thank you everyone to attend this webinar. GFB Onlus is a patient organization that is giving support from 2013 to the research and LGMD community. And this year we are very happy because we are celebrating our 10 years on the road. So, and we can be not happier, we can be even more happier because at the beginning when we start our association, the idea that we're going to have some clinical trial or some approach to our disease, it was just a rare possibility. But now, during this 2023, we are assisting to different clinical trial starting points, such as those ones from Charitè with sarcoglycanopathies, dysferlinopathy, and other programs. We also attend to the starting of the Atamayo programs with gamma sarcoglycanopathy and LGMD-R9, and also AskBio. So we are having this explosion and we are very happy. And today we are even more happy to present and to have the approach of another proposal that is arriving from Germany and Dr. Spuler, momentarily for alpha sarcoglycanopathy. Dr. Simone Spuler and her team, with a combination of different molecular techniques, have reached to correct some of the most frequent forms of alpha sarcoglycanopathy. Dr. Spuler is a neurologist and led a working group of 20 young researchers at the Charitè University, specialized in muscle dystrophies and muscular dystrophies. And her group is also part of the Max Delburg Center. Dr. Spuler studied medicine at the University of Würzburg, showing some interest to the things that we didn't not know very well, so that she can unravel it. She started to study multiple sclerosis and doing the PhD on that. And then she started a period that she goes to the US and come back to Germany and go again to the US, where she started to study muscle disease and muscle dystrophies, and where she learned some techniques that they still use today for the disease. And the thing that motivates more she and her team is the difficulty of this disease. She wants to demonstrate that there is a hope and there is a way to treat our disease. And moved for this challenge, she is presenting today the clinical trial that they will start at the end of this year, that to treat some mutations of alpha sarcoglycanopathy. So we are more than happy to have today the presence of Dr. Simone Spuler presenting this potential therapy. Thanks to be here and very welcome, Doctor. So the floor is yours.
Well, thank you very much for this very kind introduction, and thank you also very much for the invitation. I'm very honored to speak to you. And now I will share the screen, and please give me notice whether this is working.
Yes, we can show it.
Okay, so then I will go to full screen. No, wait just a second. Presentation mode. Is this okay?
Yes.
Yeah?
Yeah.
Okay, fine. So as I was introduced, I'm in Berlin with my group, and in Berlin we have a wonderful situation that the institute where my group is located is in between a very large university hospital called the Charitè University Hospital and a Center for Basic Science, which is called the Max Delbrück Center. And we, being there in the middle, we sort of can profit from the basic scientists as well as from the clinicians, and so we think we are in a quite lucky situation. We are talking about translation, and I brought you this cartoon, which I really like a lot. And now with your 10th anniversary, you probably understand very well what I'm talking about when I'm talking about the adventure of translation. So here you see this incomprehensive machine where you put in a lot of resources. And here it says, when you can see this investment, meaning money, and on the other side, the research, and you are pouring in a lot, and then it goes through this machinery, and people are sitting there wondering, you know, whatever comes out of it. And then you have these little drops down there, and the society, or this lady sits there, and it says, well, you know, research doesn't just translate itself. And then the question, why it doesn't? But this is pretty much what it is. And translation is a long road and a difficult endeavor. Okay, so you are familiar with muscle disorders, and you know that there are all kinds of muscle disorders that can be local. We are quite aware that during the COVID pandemic and people who had been on long-term ventilation, there is a severe problem of atrophy of the respiratory muscles. There can be isolated problems in swallowing. There can be generalized muscle weakness in the context of other illnesses like cancer. But then the diseases we are here most interested in are the genetic diseases, the muscular dystrophies, which are taken together not that rare, but each of the subtypes is a rare disease, and one really needs to fight for some attention for these diseases. And you know, this is what I think is wonderful, that you took up to the effort to do so. What we are working on, and you will hear a little bit of a different approach than you know from Sarepta and from other groups. So I was very intrigued by the fact that in 2010 it became clear and absolutely clear that muscle only regenerates by its own stem cells. So-called satellite cells. This has been suspected for a long time. Actually, the person who discovered the satellite cells, or the muscle stem cells, the first time, he located the cell as on a little bit different location than the rest of the cell nuclei. So it's here outside of the sarcolemma, and this is why it's called a satellite cell. And in the very first paragraph, the author wrote in 1964, he wrote, maybe we found the cell that is actually contributing or doing muscle regeneration, which has been a vexing problem. Now here we are talking about 1964, and I'm sure that a lot of you who are listening today were not even born. So for 50 years and more, it took to really have proof that this satellite cell is the cell that regenerates muscle. And so when the papers were out, and then, well, obviously this was research done in very elegant transgenic mouse models. I thought we are running a large outpatient clinic, and it might be a very good idea to look at satellite cells in muscle specimens from patients and from humans, and how frequent they are, and whether they have the same capacity and the same regeneration or regenerative capacity that we knew from mouse experiments. There has been before in the 90s, and you know that Jerry Mendel was also instrumental in the early research. There has been some hope that muscle cells would be capable of repairing muscle in muscular dystrophy. And in the 90s, there was an important publication on Duchenne. But the cells, as I said, in the 90s were not characterized. And the molecular profile of these cells and the exact characteristics was also not known. So this took another 15 years until one had the knowledge about the transcription factors and all the other factors that really make up the muscle-own stem cell. When you put these stem cells into culture and try to expand them and to get more of these very valuable cells, these cells divide and become more numerous. The problem is that this division and the further aging of the cells is really a very fast process. And this is why it has also been very difficult and not possible until now to use these cells for regenerative purposes. So here you see that here it says in the very early stages, when the cells are small, round, and quite nice, they have a regenerative potential. But when they grow older and they sort of modify their shape, they become larger and more extended, then they lose their regeneration potential. And because this process is quite fast, it has only been possible to get hold of these late myoblasts, and they do not regenerate muscle anymore. And even further differentiated cells, then they start, as muscle does, to fuse, to have many nuclei. This cell is then called a myotube, and here the regenerative potential obviously is also completely lost. So when we had started our research on this, we thought our aim would be to increase the number of divisions of these very early cells, get a lot of these cells, and preventing the cells from growing older or from aging, to make this shift in morphology. And for that, we invented a new method to isolate the cells and also to propagate them. And this is shown here. So the traditional way to isolate muscle stem cells is to take a little piece of muscle biopsy specimen of a few millimeters in size, and then to take a mixture of different enzymes and to isolate and to treat the muscle tissue with this enzymatic cocktail, and then you will get a cell suspension. And the problem with the cell suspension is that you isolate other cells than muscle cells, because there is connective tissue and there are blood vessels and there are some fat cells. This can all be in muscle, so this is not a pure cell suspension that you get. And these enzymatic digest also activates the satellite cells so that they rapidly grow and grow older. So we thought, when the muscle biopsy is taken from patients and donors, we should avoid any of this treatment of these enzymes, and we should, after the biopsy has been taken and the satellite cells have this signal, oops, we need to repair something, so they get activated, that we should stop this process again and try to calm them down. And we do this in a way that we place these small fiber fragments that were manually prepared. We place them for an entire week into the fridge. So it doesn't sound very magical, but it is in a way, because the cells in the fridge, they stay there for an entire week. And in this environment, the satellite cells go back into a quiescent state, meaning so they lose this activation problem. And after 1 week, we take these fragments out again. We place the fragments into an incubator, now with nice 37 degrees, and we wait. And then we can see that the beautiful cell populations that are young and divide for a long time can be obtained. And the other nice thing about this procedure here with the fridge is that cells that are not muscle stem cells, they do not survive this. They cannot be kept in the fridge for a week. So meaning that after we have when we look at these populations, we see very pure muscle stem cell populations, and there are no fibroblasts, and there are no endothelial cells or other cell types. And so we thought, no, this is cool in several ways. And we filed a patent for this, and this worked out. And so this is now like the basis for running trials with these young and nice muscle cell populations. And this is an example of how beautiful they look. Here you can see a marker, this is called Ki67, but it really doesn't matter. It's just an indication that these cells are still dividing, that they still have the capacity to divide, and that they are actually muscle cells. And this is being shown by a molecule or by a protein with the name of desmin. So you have here desmin positive small cells, and they can divide. And this is the type of cell population where we believe they will regenerate muscle just nicely. And we haven't started the clinical trial yet, as you know, but we have shown in mice that do not carry an own immune system, so they would tolerate human cells. And we have shown that when we inject these cells, they make nice muscle fibers in this mouse muscle. But in addition, you know, this muscle fiber is wonderful to see that. But in addition to retain and to recover the regenerative capacity of the cells, we need to show, or we wanted to see, whether new muscle stem cells were also made from the cells that we injected. And I think indeed here you can see that in the satellite cell location or niche, you can see new stem cells. And this is the very important finding that so the new muscle fiber, even if there is some strain or destruction of this fiber at some point in life, will be able to regenerate itself by the fact that new muscle stem cells have been made. And then, of course, we had to re-injure this or to even prove this. And it has been shown and demonstrated that this became true. So this is truly regenerative muscle. And when we had found all of this out, it was just about the time when the CRISPR-Cas gene scissor was found by Emmanuel Charpentier and Jennifer Doudna. And you have heard about this, and they received the Nobel Prize in chemistry in 2020. And we were so excited when we had the technique for the muscle stem cells in our hands that CRISPR became available, because then we thought, okay, now it's the time where we can start working on the stem cells of patients who are suffering from muscular dystrophies. And one of the difficult things in working with stem cells from diseased muscle is that during the course of a disease, of a muscular dystrophy, healthy muscle slowly becomes replaced by connective tissue. And the connective tissue cells, they are quite aggressive, and they like to take over cell cultures where people or researchers are mostly interested in the myogenic cells, in the muscle cells. So we were wondering whether a patient, when we take our new method with the fridge and all of this playing around with the cell activation, whether we still would be able to receive a pure cell population from patients who are suffering from muscular dystrophy. And here I have introduced the molecule desmin, which indicates a muscle cell. And you can see that with our technique, we can get 95% pure muscle populations. And I think this is very important to even to think about gene editing in these cells. If 95% would be connective tissue, then we wouldn't even need to bother to try to do any repair. But so the new cell culture technique was sort of the prerequisite of trying to do gene editing experiments. So there has been a family who is compound heterozygous in the alpha-sarcoglycan gene. Compound heterozygous means these patients have two different mutations, one from the father, one from the mother. And one of the mutations was here in the exon 2, and the other mutation was in exon 7. And taken together in the children, these children showed first symptoms of muscle weakness when they were around 8 or 9 years old. So when we took up a project like this, this definitely wasn't something that we just could do overnight, and it's a project that took us couple of years, if not more, to decide which mutations to work on, because there are a lot of other mutations as well, as you know, and to make good decisions about which mutations should we put our effort and money and time in. So we thought it's important to check whether a mutation is just a very, very private mutation that is not shared by anybody in the world, or whether the mutation has been reported in papers, and there may be, you know, dozens or maybe a few hundred patients carrying the same mutation. So the two mutations that we identified in the alpha-sarcoglycan family was here the alpha-sarcoglycan 157. This means the locus of the nucleotide is the position 157, and the other one was position 7048. And for both of these mutations, there existed papers that showed there are families around in the world who are carrying exactly the same mutation. So this personalized medicine would not be for a single person, but it would be for a group of people. And I think this is important when it comes to very, very specific, correct mutation work, and looking for you know, this much more detailed than the entire gene. It also turned out that the CRISPR-Cas9, which is like this, what we call a gene scissor, is also a very good target for a development that came out of the classical CRISPR-Cas9. And this is called a molecule that was derived from CRISPR-Cas9 and is called the adenine base editor, or as we abbreviate this, ABE. So the ABE differs from a scissor in that fact that it doesn't cut in the genes, but it just takes away with a little forceps, takes away one molecule from the gene strand and exchanges this single molecule with another molecule, and it leaves the gene strand intact. This is very nice for the cell, and it's very nice for the body, and it's very nice for clinical development, because when you cut the DNA, and then this is a very stressful signal for the cell. You know, the cell like lives on its DNA. And if you cut it, then a lot of repair mechanisms come into place that try to repair this. And some of these repair mechanisms have mistakes, and some of them are not very efficient. But taken all together, everything that is important here is sort of to glue the strand again. And all of this is not happening with the base editor. So also in other fields, not only in the muscular dystrophy field, but also in liver diseases and lung diseases, people have recognized now that the base editor is definitely going to be the first molecule that comes from the CRISPR-Cas family that will enter clinical practice, because it's so much safer and so much more exact than the classical scissor that cuts the strand. So you could say, well, super that this base editor has now been invented and found. So everything can be solved using this base editor or this ABE. But unfortunately, it's not the case, because the ABE is a little bit like a prima donna and has a lot of other requirements. It needs to work properly. And I'm not going into molecular details. This might be really boring for you, but just that it's not that every mutation that comes up is eligible for using this ABE, and one has to do some bioinformatics studies first and some playing around until one can see and be sure whether a mutation is eligible for this base editor. But it turned out that in the family with the alpha-sarcoglycan mutation, one of the mutations was a very good candidate for the base editor, and so we try to correct this 157 mutation. And here, these are data how the researchers receive the results. And here you can see in this line where the arrows are, you can see here with under the red arrow that there are two peaks, a green and a black one, and that the green and the black one equals both 50%. So one is healthy and one is the diseased one. And because it's heterozygous, the patients is carrying 50-50. But you want the G and you do not want the A. So after the base editor was in place, we've found 100% of the G, so the desired molecule here, and we wanted to get rid of the A. And here with the base editor, we could see that 100% was repaired, and this definitely is an efficiency that we would not have dreamt of when we started the project. But this is how powerful the base editor can work when the conditions around it are really perfect. And now we next needed to ask whether we see 100% repair, but maybe the cells are so stressed by what has been done. You need to put the base editor into the cell, and then you manipulate the cell, and sometimes cells are just stressed and then they die, although the mutation has been repaired. And so we looked at that and looked whether these cells after repair would still behave the same way that other normal and healthy muscle stem cells would do. And indeed they did. So when we did again our transplantation of our repaired cells into mice without an immune system, then we could see that new fibers were made, and these fibers now show the alpha-sarcoglycan, so they make the protein that was missing before. And in addition, and this is really nice for imagining how this could help the muscle to recover, that new stem cells that are carrying the protein alpha-sarcoglycan also are made. And this means that when it comes to another injury of this fiber, then there is a healthy muscle stem cell that is able to repair and to divide and to repopulate new stem cell niches. So I think here we knew that something very unusual up until this time had been happening. To deliver these molecules to the cells, there are several possibilities. Most of these possibilities that exist are not suitable for any for clinical use. But as we are interested in the clinical application, we needed to look for an application where we could deliver the CRISPR or the base editor in a way where it's only present for a short time, and then would be destroyed again, because you don't want the base editor or a gene scissor being around in the cell for the rest of the life. And here you all have heard about this during the pandemia, the COVID pandemia, when mRNA vaccines became available. So mRNA is now a molecule which everybody knows about, and it's short-lived, but it's very powerful. And we thought, okay, we can maybe deliver our base editor or CRISPR-Cas9 tools and letting an mRNA do the job and make the base editor in the cell. And so we ordered and we cooperated with a company who is specialized in making mRNAs for customers, just as we wanted it, and looked for the reaction of our muscle stem cells to this mRNA molecule. We didn't know whether it would be toxic or whether the cell would die right away, or whether we could even get the mRNA into the muscle cell. But this turned out to be a very nice match. So the muscle stem cells and the mRNA molecules, they just like each other. And we, of course, we were quite happy by that. And the mRNA was definitely also much better than other ways to get the instruments into the cell. And we could also find wonderful editing and changed our entire procedure now to delivering this CRISPR molecules via mRNA. And the alpha-sarcoglycan mutation that I was talking about, this 157, can be edited as a proof of concept, as our first project could be edited really nice and well. Now, the many patients here do carry two different mutations, and as I mentioned this before, these mutations are called compound heterozygous mutations, meaning that two different mutations are on two different spots in the same gene. And not every time we are so lucky that we can do a 100% repair. So the 157 mutation definitely was a mutation where everything was perfect, and there we can see 100%, but sometimes the effect or the efficacy of the repair is lower. And so we were wondering whether it's possible in patients who carry two different mutations to do a simultaneous repair of both mutations at the same time. We are interested in doing it at the same time because there is some cell stress, of course, combined with the repair, and so we didn't want to put too much stress on the cells by doing it sequentially one after the other. And so this project, this is just on the way to be published. So this is new unpublished data where we took alpha-sarcoglycan mutations at two different positions. Here it's 101 and 739, and here it's 229 and 739. And had a look at the fact whether these mutations are simultaneously co-editable. I want to point out, I will speak about this later briefly, but the 229 mutation and the 739 mutation in alpha-sarcoglycan are the most frequent mutation that we are looking at here in Europe. So these are there are hundreds of patients that are affected by these two mutations. And this is now, a very boring slide for you, but it's very exciting for me and for us, because this shows that as a single edit, each mutation can be targeted, and also very, very efficiently. Also here we are looking at 100%. So this is very nice. And then here you can read the word bystander. And bystander in the field of editing is always a bad thing. You don't want bystanders because this means that not only the target is repaired, but that there are lots of spots around that are also changed around. And this, of course, is not desirable in our context because we do not want to repair one mutation and introduce the next one. So this would not help anybody. And so you don't want to see bystander. And here you can see an example, like here you almost do not see any bystanders, this is all 0. And here you have something. And so this just helps us to optimize the approach. But when you look at the double edit, and this is also something that has not been done worldwide so far in muscle stem cells. So here we have the double edit, and the double edit works very nicely for these examples I was showing you. And here again we have this our friend the ABE, the adenine base editor, and this does the right thing on the right target. But then when you look at the bystanders, this is basically 0 and a few percent in one position. So this is very acceptable and very nice and very good to know that this double edit is even possible. And from all of this knowledge, and having also worked on other LGMD genes, and here you can see that we also work on calpain 3, and we also work on dysferlin, we also work on lamin, that these are the genes where we do have a handle that we can do very effective mutation correction. And so we plan, or we set up a clinical trial, which we call the basket trial. And the basket means that different genes will be targeted in this basket trial. So it's not a trial about calpainopathy or about alpha sarcoglycanopathy. It's about different mutations in different genes, and it's about repairing muscle stem cells. And here this summarizes this again. And if you want to do further reading, this is all well published, and I'm very happy on request to send you some papers if you feel like going into this. Of course, it's heavy molecular biology. Okay, so in the calpainopathy, we are working with a different CRISPR molecule, but all of the others are base editors, which, as I explained, are relatively safe to use. So this is called the basket trial. And we are recruiting for the basket trial. And as you see here, I put unusual information about recruiting. So we are not asking for LGMD 2D, 2C, 2E. We are asking for specific mutations, because in our technique, everything is mutation dependent. Of course, it has to be something with muscle, but from there on, it's only about the molecule that is able to repair this and to make out of this mutation again the healthy sequence. I put up these examples, and there will be a few more which I will reveal when it's the right time. This basket trial has attracted some attention. I think this is probably one of the reasons why Carl has contacted me. So Nature Medicine reported on that as one of the 11 clinical trials that will shape medicine in 2023. And this is what we are planning to do. So we take a muscle biopsy from a patient, we expand the cells by our new technique. We do the in ex vivo, which means we do in cell culture, we do the repair, and then we will transplant the cells back to the patient. And the clinical trial will start end of this year or early next year. So this is planned. And of course, we have more hopes. If we can correct a mutation in muscle stem cells, then definitely the next question comes up. Yeah, but it's not so many muscles that you can reach, and will that be effective for a patient? And is this good enough? Now, for today, as we have nothing yet, I think it's definitely good enough. But we also would like to apply this sort of gene correction to the entire body at some point, and this is basic research. But we have made new mice, And these mice, they carry the human gene. So we took out the mouse gene of alpha-sarcoglycan, for instance, and introduced the human sequence, so that with the same molecules we are using here on the cells, the same molecules can be used to be introduced into this new mouse model, and we can see whether in the living organism we can do a precise gene correction. This project is ambitious, and we don't know whether this is ever going to work out, but we know that it takes perseverance, it takes patience, and it takes a lot of luck. But with all three of them, we are quite optimistic that we will at some point take knowledge that we gain now from this mouse experiments and will have a systemic therapy for patients. So, but this is nothing for 24. This is maybe something for 34. Okay, I also would like to say when we do the basket trial, there will be also a new way how we will monitor a clinical modification or clinical success if we are lucky. And here we are working with an app that we made that is specifically designed for patients with muscle disease, knowing that classical fitness app is absolutely not suitable and isn't the right thing to guide patients with muscular dystrophy. And here we have something that is called Sumus, and it helps to digitalize a patient outcome, and can through a little bit complicated algorithm measure how the force and the strength and the acceleration of the arm. So it's here focused on upper limb, can be quantified, and it also tells the patient what kind of exercises to do and when it's time to rest, because we all don't want the muscle to be overused and even more exhausted. So this Zumus will also become part of the basket trial. And the Zumus is interesting because it does improve patient assessment as compared to the classical neurological exams that patients have to go through, but they are very rough and do not give a very good estimate of daily life activities. And it also saves time, and of course it saves money if patients do not have to travel to centers all the time to get an exam, but can do so just by digital interface. Yeah, and this is the group. This is my research group, and here we have Helena Escobar, and she set up the CRISPR in the lab for, she started it, and she said, look, Simone, I would like to work with you, and I do CRISPR. And this was like in 2015 when this was super new. And then we have the cell people. So here we have the people who are working on the stem cells. And now most of the group is now interested in the gene editing aspect, of course, and the mRNA story was developed by Christian. Yeah, and so this are us. And we got funding, quite nice funding from the Federal Ministry in Germany, from the German Research Foundation, from private foundations, and then from the large Helmholtz Society, which is the largest research organization in Germany, as well as some local Berlin funding. And with that, I would like to stop and to ask you to ask any question you would like.
Perfect, very nice presentation and very interesting things, all of them. So, and we have to get some questions that some of them you already answered, but maybe I just go through it because like this it's very clear for the people what your answer is and what the possibilities of this therapy will be. So from Germany, we have the question, that it's a general question, but if it is possible to stop the disease progression, and if this disease in the people who are already very severely affected, if this therapy can be effective or not.
As I said, it's really dependent on whether we can get enough muscle stem cells to do the repair. And if a disease is very much advanced, there is a limit to at a certain point there are not enough cells that one can collect. If there are no muscle fibers anymore, then there are no muscle stem cells either. To collect these muscle stem cells, it's advisable to do this at an early stage. But we haven't if we don't get cells, we cannot do any repair.
Mm-hmm. Okay. And then from Canada, they say if this is some kind of some stem cell therapy, or it should work with some donated cells. Well, you already said that this is from the muscle biopsies, but it should be some kind of stem cell therapy.
Yeah, so donated cells. So here, as I show here, it's autologous, meaning that the cells will be given back to the same patient who donated the cells. If the donor is a different person and could be a healthy person, then one doesn't need to go through the trouble of gene editing, because then the cells are healthy. But we have learned that these allogenic donor cells just do not engraft in muscle, and then you have to give additional medication which are not well tolerated, but these approaches have simply not worked.
Okay, and then maybe related or not, that is the heart, that is a muscle as well, and it should be difficult to have this some kind of cell to regenerate it. So this is a proposal for the moment, just for skeletal muscles, and if it is now for the moment just intramuscular, no?
It's intramuscular injection of the cells, yes. This is here the basket trial is not a systemic approach. This is something,
Okay.
We will be working on, but the basket trial is a local approach.
And another question from Italy is that we have seen that these clinical trials are most of them developed by companies or big companies, and do you have some support for this trial, or if you have started talking with some companies, or it's just for the moment you're
yeah, so we are in discussion with a couple of companies, and we also started a small startup to ensure financing, and also to support any way to make this available to all patients. We are talking here about a clinical trial, a small clinical trial, but if something like this will become or should become available, then one needs to go through market approval and some other steps that are lengthy and very expensive. And here we definitely need support by Big Pharma. And they are definitely interested, and we are talking to them. Yeah.
So some people also ask they are interested in this small clinical trial. So they said where it will be located, or if you can explain a little bit more about the pipeline.
Yeah, so the location where the trial will be conducted is Berlin. and we will here, if we need to take a muscle biopsy, we need to see that we get the cells. But we know that the mutations I was talking about, that they are repairable. So it's not a project about trying to repair a mutation, but here we know it can be edited, and we just would need cells. And but this is done in Berlin, and patients who are interested can contact me by mail, and they definitely will get an answer.
Mm-hmm. And another question is, how long does it have to recover after after the injection? So what should be the recovery that we should expect of that?
We expect the transplant to be transformed into muscle fibers within 8 weeks. And this needs to be tested, of course, because this has not been done in patients. But we would think that we check for new muscle fibers after 12 weeks, and we check for improved strength, which also, of course, is very important clinically, much more important for the patient, that the muscle has regained some strength rather than some histology or some academic stuff. After 6 months.
Mm-hmm.
And we would like to measure this in a classical way, how we have done that for many years, but we would like to implement this new app, the Zumus Watch, also in the outcome, because we think that the daily activity will tell us much more than a single examination in a doctor's office.
Mm-hmm. Okay, and as a future plans, or just imagining these kind of therapies to have these cells and to treat it ex vivo, and this could be also useful, for example, to combine with the current gene therapies.
Yeah. So here we are not facing any anti-AAV antibody responses or anything. So here I would sort of imagine a scenario where you have a Sarepta trial, and the AAV virus with the additional cDNA will be given, and then hopefully some effect will be there. But one could sort of boost single muscles with our approach. Let's say muscles that are very, very crucial for daily life, and this could be like the finger muscles, finger flexors, or the other muscles, biceps or foot muscles, or you name it. So this could be there would be no interference between the approaches, and I think this also makes it powerful. And if you look at the entire field of medicine and internal medicine, then you know that someone with arterial hypertension also is usually treated with 2 or 3 different drugs and not with a single drug. And this, of course, will be true for muscular dystrophy too. It's of course will not happen that there will be a single way and then everything is treated. So here a combination of different approaches that can combine and do not inhibit each other would definitely be very nice.
Perfect. So the questions that we received, it's till here, and I think we have some questions also in the chat that I could not read. If Claudia or someone want to read the questions of the chat, if not, I go through it, or Lushika, I don't know. What is on the chat?
Okay, yes, just a moment.
Okay.
Okay, can you hear me?
Yes, sure.
Good afternoon. So I read some questions that we have received. So the first one, okay, so the first one is, can this approach be used in large muscles, for example, proximal muscles, and can a sufficient number of cells be regenerated to restore proximal muscle function?
This, of course, needs to be shown, but we would say that there are muscles on the shoulder girdle that are, although they are large, they are approachable.
Okay. And then another one, would the homozygous variants, for example, C197T measured than A, be technically easier than a compound heterozygous mutation for gene editing?
Yeah, yes. So homozygous mutations are a little easier because you don't have to worry about two alleles. Yeah, it's easier, but as we thought it's very interesting for the many people who are suffering from compound heterozygous mutations to have a look at whether they are approachable, and they are, but homozygous mutations definitely are easier.
Okay, next one. And then someone is asking if there could be any hope for curing LGMD2D patients. Yeah, okay.
Yeah.
So, yeah. And then another one, how can we participate in the basket trial? How to check if we are eligible for it?
Well, first of all, you need to have your diagnosis, and usually you get this from your treating neurologist, and you should have your genetic report. So I know from experience that patients are not quite used to the fact that, you know, we ask for the specific mutation. You know, sometimes it has been enough to know roughly, you know, what kind of muscle diseases this is. But in the genetic report, it's always written the exact mutation, and you know, I listed the mutation that are eligible. And so if, you know, anybody checks the report and see whether one of the mutations is there, they can contact me. And as I said, you know, the right mutation is of course a prerequisite, but also the fact whether we can get the cells.
Okay, thank you. And then there is a reference to a site, and someone saying that PubMed.ncbi alludes to presence of C197T as a founder mutation. And then there are some SGCA. And when do you anticipate expanding to future mutation? So I don't know exactly. It's very technical.
A matter of man and woman power, and a matter of money, because, you know, tackling a new mutation is a project that takes a while, and it's not only about, you know, can you repair the mutation, but the most of the work goes into the fact, are there so-called off-target effects? Are there other things that are happening in the cell or in the genome that are not wanted? And this is the question that really makes the safety issue of everything so important. I didn't elaborate on this, but this is a lot of work, and so it's a matter of work and money.
Okay, and the other one is the difference between, they're asking for the difference between Myopax and Myopaxon used by Myogenica.
I didn't understand the question.
Okay, maybe I can read, maybe, okay. Could you explain what's the difference between Myopax and Myopaxon used by Myogenica in the US? I guess the approach is different, but uses the same stem cells. This is the complete question. I don't know.
I can't really say what they are exactly using, but so I just know our product.
Okay. And then in addition to SUMUS, will you also include other functional assessments into your trials? For example, 100 MWT /NSAD/NSAA/S6 MWT and other.
Everything. Yeah, of course. We do all of that, but in addition, we do SUMUS.
Okay. And is this therapy possible for another sarcoglycanopathy like LGMD2C?
Yeah. So if there is a right mutation and there was you know, enough time, support, and work power to work on the mutation, every mutation is eligible as long as it's in muscle. Yeah, so it doesn't need to be expressed in muscle stem cells, but otherwise there is no restriction on the gene. It's a matter of the environment of the mutation, whether it's eligible or not. And then, of course, we are 20 people. You have seen the picture, and there's only so much work we can do at the same time.
Okay, thank you. How do you plan to get the stem cells to the whole body?
We are not planning to get the stem cells to the whole body. We know that this is something local, but we are still looking at diseases that cannot be treated at all. Sometimes it's fine to have 2 or 3 muscles that are functioning better than they have before. And as I said, this is our approach here in this slide. This is how we want to get the CRISPR molecules to the whole body, but this will be without the cell. It will be the scissors, and the scissor will be injected into the whole body.
Okay, thank you. And then someone is asking the address where they can write to get more information about the trials and how to submit as volunteers.
So I'm sure that through you, my email address can be, I'm happy to receive any emails. I didn't put it here on the slide, but maybe I can do so. Just a second.
Okay, thank you very much. No, ce n'è ancora una.
So, now I'm sharing again. Okay, now you can see that. So here, I don't know whether you know Alan Johnson's Sirleaf, but I think this is very nice, this sentence. So here it's simone.spuler@charité.de or mdc-berlin.de. On both email addresses, you can reach me.
Okay, thank you so much. And then the last question is, how much funding would be needed to target homozygous C197T major than A? Because he's saying that he or she, I don't know, they are saying that some families are desperate enough to fund some research themselves.
So the person who is asking the question should write an email to me, and then we do some bioinformatic analysis to see whether there's anything on the theoretical part that can be done, and if so, then we can discuss whether we can take up the experimental work. But the bioinformatic analysis, of course, is free.
Yeah, okay, thank you so much. And so if there aren't any questions, any more questions, I think we can finish here. And thank you so much, Dr. Spuler, for being with us. And so we really appreciate it. And thank you, Carlos, and thank you so much to everyone who joined us today. Thank you. Bye-bye.
Okay, bye-bye.
Thank you.
Bye. Thank you. Bye-bye.